Yes, good evening to all the participants. So, welcome to the session 9 on macromolecular structure analysis. That is specifically on we discuss on protein structure prediction and protein structure visualization. Okay, so before going to protein structure prediction and visualization, so can we go across the last session topics? Yes. So, what are the last session topics we have discussed? Very good. Once again from Prema Madam. That is, she has mentioned that we have discussed on distance-based methods in molecular phylogeny. Especially, we discussed on unweighted pair group method of averages. So that in the distance-based method, okay, how to construct a phylogenetic tree. Okay, so here we have seen three steps in UPGMA. In overall, we call this UPGMA, we segregate this in sequential clustering method. So we segregate this type of distance method as clustering method. And even we have discussed like if you are provided with sequences instead of distance table, then how to form a distance table from the sequences. We started about that. And after solving a problem, we also discussed like if the sequences present in row and column are not matching, then we have to check and we have to frame the table accordingly. Okay. And we also discussed something on the other methods like neighbor joining, maximum parts, that is character based methods. We have discussed maximum parsimony and maximum likelihood. We also distinguished about these methods, and then we discussed what is bootstrapping. Bootstrapping is nothing but a random sampling for checking the robustness of the trees. And we also discussed on the tools present in programs present in Phylic. Other than practicing in Mega 7, we also discussed what are the tools present in Phylic package. You remember 35 programs are available in Phylic. So for each and every method, you have unique programs in Phylic. Okay, so we also discussed about that. And if you download and install the software, we can find doc, src, exe folders. So inside exe, you have 35 programs. If you want to study about the Phylic particular program, you can go for doc, for subfolder present in Phylic. Okay, now we after that we just seen various tools like ITOL. In ITOL, how to see the tree of life can be bacteria, virus, algae, even for mammals. So for all this, we can able to see the tree of life for multiple species. We get a kind of a radial tree. Other than this, we also discussed about the various outputs of phylogenetic tree. You remember? Dendrogram, soupogram, curvogram, other than phenogram and cladogram, we also discussed about this and how to distinguish evolutionary trees from phylogenetic trees. Phylogenetic trees are constructed basically on distance and characters, but evolutionary tree is constructed not only based on the characters and distance, but also we take time in consideration. Okay, so this is what extensively we discussed on phylogenetic trees. Now let us come to today's session. Okay, and before going to today's session, if you have some kind of internet disconnections, okay, if my voice is not audible, kindly let me know immediately. Okay, so I try to change kind of internet. Okay, okay, let me continue. So we are going to discuss on macromolecular structural analysis. That means even we discuss about the other molecules, peptide molecules, and even other molecules. Now let me start just with the protein structure prediction. So the macromolecule we majorly discuss on protein. Why we discuss on protein? If you take any diseases, for all the diseases, proteins are the important targets. So one has to use a target receptor from protein macromolecule. So if you see, then what are the micromolecules? The micromolecules are drugs, which are generally a chemical compounds. We can also use synthetic compounds as drug. An example is paracetamol, aspirin. So these are synthesized, but these can be used for as a drugs, okay, for treating fever and treating body pain, isn't it? So, chemical compounds, synthetic compounds, and whatever compounds derived from biological sources can be bacteria, virus. So, all these things, okay, bacteria, virus, algae, fungi, all these things can also be used, okay, as the micromolecules, and even DNA and RNA can also be discussed. Like if you see in micromolecules, we see almost all the biological molecules, including lipids, carbohydrates, etc. All these molecules, whatever I mentioned, can able to bind with macromolecule like protein. So macromolecule, obviously, no doubt, it is a protein. But in case of antigen antibody interactions, in case of antigen antibody interaction, okay. Then obviously, mostly the antigen will be a larger molecule. If then that can be considered as a macromolecule. An antibody, just because of smaller size, it can be considered as a micromolecule in that case. That is an exception in protein-protein interactions. Otherwise, majorly you can find our protein is the only macromolecule used in almost all the studies in all the types of biology. And if you see this kind of a protein structure prediction, okay, so we can able to see there are just I mentioned four levels of protein structures, but we also have five levels. The between secondary and tertiary, we have a class called super secondary. Super secondary is the third level of classification. In primary structure, we have only the amino acid sequence. And if you see the secondary structure, we can find in the form of alpha helices, beta sheets, loops, turns, coils. Okay, so these are the other kind of secondary structures. Even in alpha helices, one can able to find the types like left alpha helices, right alpha helices. Okay, so pi helices, 310 helices. So likewise, we have the types of alpha helices. And beta sheets, we generally have two classes. One is parallel, the other is anti-parallel. So this is something about the classes of beta sheets. And we also have loops, turns, and coils, which comes under category of secondary structure. And if you see the super secondary structure, in super secondary structure, we find the protein is present in the form of motif. If you see the motifs, we have motifs like beta, alpha, beta. Here, whatever I can tell like an alpha helices are represented by alpha and beta sheets are represented by beta. So in super secondary structures, we have a combination of this secondary structure. So for example, one such motif, what I've given for super secondary is beta, alpha, beta motif. And then jelly roll motif, creaky motif. We have plenty of motifs like this. Okay, I just given few examples for motifs. And after this, if you see that the fourth level of structure is tertiary, in tertiary, we have a combination of motifs and we find the protein structure is present in the form of domains. So in tertiary structure, we represent in the form of domains and super secondary structures, we represent in the form of motifs. What is domain? Actually, domains are nothing but a chain. So actually, instead of domains, I should have mentioned like domain, a single. Okay, if you see in this case, we may have a chain A in a PDB structure. The 3D structure of a protein is generally present in the form of .pdb extension, that is file name .pdb. PDB stands for Protein Data Bank. That is the 3D structure of protein. Fine. Okay, most of the databases have these extension .pdb. Then what are the other extensions we can find? We can find .pdbqt and we find even other formats also. Okay, but most probably we find like filename.pdb. Yeah. And if you see this kind of a tertiary, I told you we have in the form of a single chain, can be a chain A or chain B, and that's what we call it as a domain. If you go to the final level of protein structure, we call that as quaternary, and in quaternary we have multiple chains. So here you have only one chain, but see here, according to the colors, so this is dark shaded, so this is one chain, and this is somewhat light shaded, this is chain B. And again, when compared to this, you have slightly shaded, that is chain C. And here you find no shading, that is chain D. Just because we have four chains, we call this as multi-domain. So in multi-domain, we can observe in quaternary. So the difference between tertiary and quaternary is like in tertiary you have only one chain, but in quaternary you have more than one chain. Can be two, three, or four. Okay. Generally we have two, four, eight, something like that. Okay, in multiples of two. Except in few proteins we have triplicates also. Generally we have quartets. Okay, and if you see this kind of a major, that is, what is a 3D structure of a protein? If you get a question, okay, what is it what is the 3D structure of a protein? Then you can tell not just the quaternary is the 3D structure. Okay, we also find whatever for a chain A, which we call it as a domain, is also a 3D structure. So 3D structure of a protein starts from tertiary. But in quaternary, you have a protein in the form of a stable complex that is binding with multiple chains. Okay, so it starts from here and it can finally ends with a final level of structure called quaternary. Okay, now let me go to the composition of protein. So just before going to the slide, can I ask you a question? So you can try to interact with me. What is the major composition of a protein? Participants multiple. Just admitted. What happened? Okay, participants. So you just use that link. Okay, first to use that link and you can try to. Okay. Now I was explaining you something like yes, very good, but still this is not a correct answer. Very good, you're alert. And even I found many participants are interacting now. Those who didn't interact earlier. Very good. I appreciate your involvement. But the question I just typed, what is the major composition of protein? Of course, you are coming with answers. But so this something general. Whatever you mentioned, amino acids or building blocks of proteins. But what is the polypeptide is a combination of amino acids. Still, so I'm telling amino acids are the building blocks, and a combination of amino acids we get like a peptide, polypeptide. What is it, ma'am? Uh, no, but the answer looks like silly. That's not a correct answer. Even nitrogen, hydrogen, carbon, everything, whatever mentioned, or atoms, isn't it? CHON. So in protein, we find also N. Okay, so whatever Bhagyalakshmi has mentioned is right. But what is the major answer? Can I type the answer? You itself find silly. But this is the major composition. We have to accept it. You know, in our body, more than 70% is filled with water. Not only in the earth, we find major portion as water. Even in our body, in blood, we find major composition as water and even other proteins also. Okay, more than 70% in volume. But if you take the major composition, the building blocks of a protein, then we tell it is an amino acids. No doubt in that, because the amino acids are we can find nearly 20 amino acids in a protein. Okay, so we are not bothering about the other amino acids like ornithine and other non-essential amino acids. We focus only on the 20 amino acids which can build a protein structure. If you see the
Excuse me,
yes, you can ask me.
sir, I cannot understand the how major composition of amino acid is that is water.
Um, Simply you have to check in internet through Wikipedia. Like If you just see the composition, okay, like in the protein, like so what is what are the composition of a protein actually other than amino acids? So I can also ask this question. Other than amino acids, what else we can find in protein? Can I ask this question? You can try now. Other than amino acids, what else we can find in protein? So later I can try to come with an answer. I justify my answer. What are the molecules present other than amino acids in protein? But one thing, I think those who ask me, you are intellectual in asking this question because even if you download the 3D structure of a protein, we find only few heteroatoms, isn't it? This might be made me made you to question me. Good, nice interaction. But let me explain this also. What are the other components present, other molecules present? So carbon, hydrogen, or atoms? No, this I can't accept. I am not asking the atoms, but molecules. Very good. Iron. We find a complex iron molecules in case of hemoglobin protein. So now we are bringing a complex iron molecule. What else, madam? What else other than carbon, hydrogen, nitrogen, oxygen? So we can also find. So if you take the molecules like iron, you not find just as a metal, but you find the form of a molecule. Like iron, we can find zinc molecules. We can find calcium binding proteins. Okay, and what else we can find? Selenium and any other zinc, calcium, selenium. So these are the popular binding proteins, isn't it? We can find this, and we can also find the the all these zinc, calcium, selenium, and even if some drugs are binding with the proteins, all these things are showed as HET-ATM in PDB structure. Okay, yes, very good. Phosphate ions, or sometimes even carbohydrates binds because carbohydrate binding proteins are there, lipid binding proteins are there. So all these things are considered as HET-ATM in 3D structure of a protein. But only atoms present in protein or amino acids. Okay, whatever mentioned as atom is amino acid, but heteroatom takes the other molecules like lipids, carbohydrates, phosphate and, ions, and other kind of ions, zinc, calcium, selenium, iron, etc. Okay, so these are the major thing you can find in 3D structure of a PDB. But okay, like when you ask about the water molecule, okay, water molecule also comes under HET-ATM. If you see in most of the protein molecules, except iron binding proteins, we find iron. Zinc binding proteins, we find zinc. So very few proteins have this kind of specific molecules. But if you take almost all the proteins, like in our body, whether you justify that in our body we have more than 72% of water. Those who ask me a question, can you justify my answer? Yes or no? In our body, yes. If so, then where is the water? Where the water is stored in our body? If we have more than even we see so many kind of aqua cod advertisements in our TV that they're telling mentioning about the water composition of our body, where it is stored. Even if you take a blood is a protein. We have so many cells, and in the cells we have so many proteins, surface proteins. We have intracellular proteins also. Obviously, one have to remember. Of course, in blood we have much, but even in the other proteins, other than hemoglobin, we also find in other proteins also. Okay, so this is something general what I learned in anatomy. Okay, now let me come across in amino acids and protein. So if you see an amino acids in protein, which distinguishes each and every proteins, isn't it? So like however you find iron in hemoglobin, we can also find iron in myoglobin. Myoglobin, of course, a muscle protein also linked with the blood. Okay. So this but the myoglobin function is different. Hemoglobin function is different, even though it has a iron molecule, and even the structure of iron molecules varies in hemoglobin and myoglobin. But if you see the structure, overall structure, just because we have a globin fold, both hemoglobin and myoglobin shows similar structure. It is difficult to distinguish these structures. Hemoglobin is a blood protein meant for transport function to transport oxygen, okay, while inhalation, and to exclude CO2 while during exhalation, we use hemoglobin, isn't it? And if you see the myoglobin, of course, even though you have an iron molecule, but majorly you are not using this iron molecule for transport, but it is for some other purpose, like for building the structure, for maintaining a structure, to keep it intact of myosin and the fibers, okay, muscle fibers. So you have a different functions with the protein, okay, even though have some same kind of iron molecules are present. Okay, so one can able to find a different function. So not only that, even though hemoglobin and myoglobin shares a common structure, the amino acid sequences present in hemoglobin and myoglobin are different. Amino acid sequence present in hemoglobin and myoglobin are different. So likewise, you can also find variation with respect to each and every proteins. Then I just mentioned we have 20 amino acids in a protein. So these 20 amino acids are categorized, okay, based on positive and negative charges. In negative charges, we have aspartic acid denoted by single letter D. As a bioinformatician, one should immediately remember the single letter characters of the amino acid. Okay, aspartic acid D, glutamic acid E comes in the negative, and one also should remember the classes, major classes. Like one is classifying based on charges, positive and negative, and the other is classifying based on polarity and uncharged. Okay, if you see the polarity uncharged, we have serine, threonine, asparagine, and glutamine. So the single letter words are mentioned. And if you see the hydrophobic side chains, we have AVILMFY. And so we can, whatever is not coming under either in all the four classes, we are grouping it in special cases like cysteine, glycine, and proline. So this, if you there is a much information, but I can't able to teach you the protein chemistry at present. I have to take only the basics to cover the bioinformatics. So stepping into the next slide. So in this slide, we can able to see an amino acid is interacting with an amino acid through a peptide bond. The peptide bond is what we highlighted in yellow. Okay. So you can see this. This is here exactly you can find. This is not a peptide bond, but when an amino acid is binding with an amino acid, okay, then obviously one water molecule is liberated. So only one water molecule. What is one water molecule? HOH. That alone will be liberated. So you can find here in the form of a peptide bond. So these two amino acids are merged together through the peptide bond. So one water molecule is dissipated. Okay, so this alone you get as during formation of peptide. And if you see the globin family, just I explained you, we have various globin proteins. Like even in hemoglobin, we have hemoglobin A chain, B chain. When hemoglobin is taking oxygen inside, we call this as oxyhemoglobin. And when it is exhaling CO2 outside the body, okay, then we call it as a carboxyhemoglobin. So the oxyhemoglobin is a structure, different structure. Carbon dioxide hemoglobin has a different structure. But I mean telling you have a different structures, but the structure looks similar. I mean the sequence variation you can find. So hemoglobin have multiple chains. We have oxyhemoglobin, carboxyhemoglobin. We have myoglobin, and these are the proteins are generally found in mammals. But see here, the next protein what they mention is leguminous hemoglobin. Leguminous. Where you can find this protein? So obviously from the name, leguminous is from leguminous plants like chickpea. Okay, and sea urchin hemoglobin. So we have in each and every species we have a type of globin proteins. So here, if you compare the sequences, how come we can compare how, the human hemoglobin blood protein with the chickpea protein, not a chicken pea, chickpea protein, whether the protein looks similar? No, isn't it? Even in the sequences, you see the wide variations. But the structure wise, because we have a globin fold, the structure is similar. This is how we can understand. So overall, you can see if you compare the hemoglobin and the plant globin proteins, you may find even up to 16% similarity. You have only 16% matches. It means that we have 84% mismatches and gaps. But still, you have a common 3D structure. You can't distinguish at all. Okay, if you see the 3D structure of a protein. Then how to see this protein? So first of all, I show you how to download a protein structure and how to visualize it. Later, I can discuss you more about this. To download the PDB structure, you remember in the first class, I was explaining you about to go to the RCSP PDB or to go to the NCBI structure. In NCBI structure, default it takes you to the MMDB, Molecular Modeling Database. Either download a structure from NCBI structure or you can download from the RCSP PDB. If you want to download specific protein like insulin, type insulin. Or you can even type the keyword. INS also takes to the insulin. Or else, if you if you know about the PDB ID, you can type the PDB ID, whatever we know. If you see this protein structure database, okay, here we have 2.25 lakh structures, okay, deposited in the database. And according to this database, okay, we are that the protein molecules deposited are derived from gold standard techniques. What do you mean by gold standard techniques? So first I take you to an example and then can explain you about that gold standard techniques. Okay, but the remaining computed, so if you see the previous one, okay, that is 2 lakh 25 is from PDB, but 10.6 lakh 8 lakhs, 10.68 lakhs are computationally predicted. You know how this is predicted? Through artificial intelligence techniques. Through artificial intelligence techniques, through the computational AI techniques, whatever predicted models, okay, these are exclusively present in AlphaFold database. So through AlphaFold model, the protein structures are predicted. And even this you can use. Okay. So some models are derived from the wet lab techniques and some models are derived from the computational techniques. Now, I'm going to type a keyword called insulin. I'm going to search the types of proteins present. If I am searching a protein, so you can find in filters like a different species. One can select a species to get a specific species. So if you see the Homo sapiens insulin, we can find almost 8,498 structures. Okay, so most of the structures are also engineered insulin or the insulin bound with a drug, something like that, insulin bind with a peptide. But if you want a raw insulin, just take the raw insulin alone in the out of 8,498 structures. So other than this, we also find a filter called taxonomy. And majorly, we have something like experimental method. In experimental methods, okay, in experimental methods, we have types of experimental methods like X-ray diffraction, X-ray crystallography. Actually, X-ray crystallography is not there for insulin, but for the other proteins, you may find this also. Other than X-ray diffraction, you can also find X-ray crystallography, electron microscopy, solution NMR. This is a type of NMR. Then what is the other type? Solid state NMR, powder diffraction, neutron diffraction. Like X-ray, you also have powder and neutron. And we have electron crystallography, fiber diffraction, solution scattering. So these are the experimental methods, wet lab techniques used to get a protein, and whatever protein derived from these techniques are mostly 100% perfect. So one can use these structures, okay, download these structures and use it for molecular docking, MD simulation, that is especially for drug design, vaccine design. One can use these structures as a targets. Generally, we download the PDB molecules as a macromolecular target and we check the binding affinity with micromolecules. Okay, Now let me complete the other filters. We can also find the filters like refinement resolution. Okay, these are DNA and RNA binding proteins. You have proteins alone, you have DNA binding proteins this much, RNA binding proteins this much. And here, if you see the refinement resolution, so the proteins are also classified based on resolution. So the proteins are classified into three types based on the resolution. So the first classification is if your protein is in between 2 angstrom to 4 angstrom. Okay, we call this as medium resolution. And if our protein structure is above 4 angstrom, then it is low resolution. If it is less than 2 angstrom, then we mention it as high resolution protein. So generally, if the angstrom is less, okay, for example, if you have 1.7 angstrom, then whatever protein structure you predict, like what is angstrom here, what is resolution here, a distance between one atom to the next atom. So if your distance is 1.7, okay, then even if you're zooming, okay, so whatever protein, if you can zoom in the visualizers, it will be more precise. Okay, if the distance is more, like if you have something like 3.8, okay, so just because the distance is more, okay, your pixel quality is not good. Like in a picture, if you take in high megapixel camera, then you get a kind of a good zooming capacity. Even if you zoom, you can find the quality is not compensated. But here, if you have a high resolution, if you have a larger distance, the quality is poor actually. Okay, so one has to distinguish the resolution of proteins like that. So if you want to download a best kind of a protein, we also have 0.521, isn't it? We have 11 base proteins. I suggest you to select this. So mostly these kind of 11 proteins, whatever we have selected, okay, or predicted recently from recent wet lab techniques. So one can use this thing, okay, actually from 1975 you find up to present so many proteins are deposited. So this is the best filter I can suggest you. After selecting this, click this refine. So obviously you can refine the protein structures to 11. So here you find so many kind of insulin proteins, whatever is desired, okay, and if it is applicable, you can download. If it is not present, then you can also go for the next resolution and you can find this because this gives you an accurate result when compared to the previous one. Okay, next we can try with 1 to 1.5, something like that. Okay, now this is how we can able to find this information. And if you click this 10.68 lakh structures, okay, so it directly take you to the AlphaFold. Okay, so it shows you the AlphaFold database. So see here, in AlphaFold we have around 9.99 lakhs, but we also have model archive. Okay, model archive. This is another kind of a technique. Okay, 69,326 structures. Very few structures are there. And whatever artificial techniques used to come up with a 3D structure of a protein, okay, so these are assessed through PLTTT scores. Actually in IIT Madras, I'm working in protein bioinformatics lab under the guidance of Michael Comia, sir. So he has come up with so many kind of models through AlphaFold. So he has deposited so many models. He also assesses the quality. So we do all these things. Okay, so in that lab. So now let me come to the next concept. Biomolecule structure determination. So if you see the protein molecules, each and every amino acid is a combination of multiple molecules. Okay. So of course CHON is there. But if you see a amino acid, obviously one can able to see an alkyl group, carboxyl group, amine group, and hydrogen group, isn't it? So, but if you take each and every groups, you have plenty of atoms, CHON. So this is going to interlink a molecule, okay, and obviously can find a kind of a bonding and non-bonding interactions between the atoms, isn't it? So actually, according to the theory, we consider each atom is simulated as one particle. Okay, so we are using actually biophysics for understanding the bioinformatics. If you see the bond stretch, okay, how a bond can be stretched, okay, so based on the bond stretch, one can able to calculate the potential energy, okay, of a bond stretch like this. This is the formula, okay. And if you see what is B actually, the B is the final, I mean, the B is the maximum length extended, and B naught is the initial length. So B minus B naught, see this is the final and this is the initial. So it has a kind of a spring-like motion. Okay. So we also consider this for modeling, talking, and simulation. That kind of, and so through this we are calculating the energy. And if you see this, this is a bond bend. Okay. For bond bend, we are calculating the potential energy like this. Okay. This is the formula K theta, theta minus theta naught. So obviously the largest angle is theta and the smallest angle which can fold itself is theta naught. And this is for bond torsion. Okay. See the moment, and you can also see the formula. So the sum of bonded stretch, bond bend and rotate along bond potential energy is what? The total potential bond energy. This is what we call it as total potential bond energy. Now let me come to the non-bonded. Non-bonded is the sum of van der Waals and electrostatic. So non-bonded potential energy is calculated like this. So what is the formula for van der Waals? What is the formula for electrostatic? So general we have a van der Waals formula like this: electrostatic Coulomb forces of attraction. Potential energy can be calculated through this. And the sum of bonded potential energy and non-bonded potential energy is what? The total potential energy. And if you see in the next slide, we can able to see how to determine the 3D structure of a protein. So in bioinformatics, okay, I told you, if you want to predict a 3D structure, so even if you predict a tertiary structure, it comes under 3D structure. If your protein is having 4 amino acid, obviously whatever you predict will have, I mean, if your protein is having 4 chains, then whatever sequence you use for prediction also is going to predict a protein with 4 chains, even though you provide a single sequence. Okay. So what I mean is that uh, the structure prediction, the 3D structure prediction starts from tertiary. And if you see the tertiary structure prediction, there are 3 methods of protein structure prediction. Number 1 is homology modeling. Number 2 is threading. Number 3 is ab initio modeling. So if you see the homology modeling, we have Swiss model, the most widely used technique is Swiss model. And for threading, we use Fiery, okay, or Fuge. And in ab initio, many people try with a ProCheck, Anolia, Rosetta, etc. Okay, so all these tools are present in XPassive Observer. In XPassive Observer, if you click the hyperlinks, it directs you to the corresponding tools. Okay, for homology modeling, threading, and ab initio. If you see the secondary structure of peptide, so before going to the tertiary structure, if you want to see further information on secondary structure prediction of, okay, so the alpha helix has this phi and psi value, and the omega value is this. So we have, we have 3 angles. So that is phi. The phi angle is present between N and C alpha. Psi angle is present in C alpha and C double bond O. And in C double bond O to NH, we have an angle called omega. So if you see this category, these are coming under classes of secondary structure. In these classes, you have the phi, psi, and omega angles vary with each other. This is how we distinguish these secondary structures. Okay, and in secondary structure of the major of a major structure, that is, if you see the proteins, the major secondary structure is present in the form of alpha helix. So next to alpha helix, we can find in beta sheets. Then one we can have the loops, turns, coils, etc. Okay. So alpha helix is the majorly found secondary structure in most of the proteins. If so, first, our objective, if you want to predict a structure of a protein, okay, one has to initially predict the secondary structure. Then only you can go for tertiary structure. Okay, if you are predicting a secondary structure, okay, then what is the major secondary structure? You can find alpha helix. To find the alpha helix, one has to see that five most common amino acid present in alpha helix are methionine, alanine, arginine, lysine, and leucine. So these are all alpha helix favoring amino acids. I mean alpha helix forming amino acids. But there are some amino acids which won't favor formation of alpha helix. What are that? The most important are proline, which we can also call it as alpha helix breaker. And the second one is glycine. Okay, because of many angles of rotation, this is also not a good amino acid for alpha helix. So if this is not a best for the proline and glycine are not best for alpha, this may contribute for beta or this may contribute for loops, turns and coils. But generally in alpha, you don't find this. And other than this, we also find unfavorable amino acids of alpha helix are serine, aspartic acid, aspargine, threonine, valine, and isoleucine. Okay, if only if you know what are the amino acids which favor alpha and which are not favoring alpha, from that we can able to conclude what are the amino acids going to form beta. If that is also not forming a beta, then it's either has to, I mean, it has to form loops, turns, or coils. Okay, so I were telling the proline is a helix breaker. So just now I mentioned about the angles phi, psi, omega. So if you see in proline amino acid, okay, we have only one rotation called phi. That too, we have a very restricted rotation. So whatever you have this NC alpha, you have a restricted rotation. You don't find a good kind of a rotation in proline. And if you take glycine, you know, we have all these three rotations with flexible. So all these the atoms rotate like you have seen the torsion rotation previously, isn't it? You have that kind of a rotation in all the three angles you find. So if you see this alpha helix, okay, so in this end, so where you find the alpha helix is going to break, okay, if a proline or glycine, you can't able to find a proline or glycine in this. Okay, so if you find a glycine, you can find it going to break an alpha helix. If you find glycine here, no problem. How about here? Okay, it just unfolds something like that. You try to break the alpha helix. So generally, overall, the proline and glycine are not favored because of this helix burns, helix bends. Okay, so and if you see only from alpha helix, one can able to go for beta sheet prediction and then the other. And now, if you see the X-ray coordinates, how the PDB structure is determined. Okay, so the most difficult task to get a 3D structure from wet lab is to get a crystal in case of X-ray crystallography. In X-ray crystallography techniques, one has to bring a protein in the form of a crystal, which is the source to produce the 3D coordinates. It is easier to bring a crystal with a small molecules. If you want to bring a drug, if you want to bring some synthetic molecules, synthetic compounds, just because of low molecular weight, it is easier to bring a crystal when compared to macromolecule like proteins. Because more protein molecules, molecular weight ranges from 10 kilodaltons to 100 and even it go beyond that. Isn't it? The molecular weight is too much. So if you form a crystal, then it is easier for us to get a structure. The steps are initially, if you pass this crystal to an X-ray diffraction onto our X-ray crystallography equipment, you get a kind of a diffraction pattern. Like you find only, we can able to find only black dots like this. Like in X-ray, how you find only the black, gray, and white. Likewise, even for a protein crystal, you find only the black, white and gray colors, the light black colors. So through these colors, okay, if you pass it to simulation softwares, okay, simulation softwares, so it can able to bring a 3D structure like this. So before stepping in, we have an intermediate step like getting an electron density maps. So through the electron density maps, you form the amino acids, you form the interconnections of the amino acids, and finally, after we are coming up with a secondary, tertiary, and even if you have a quaternary structure for a protein, you get a fine 3D structure like this. This is how a 3D structure looks like. Okay, so one can able to find only through the X-ray coordinates. Okay, this kind of a diffraction patterns. One is getting the structure through, of course, a simulation softwares like GROMACS. And if you see the methods of protein structure prediction, the first prediction is the first method is homology modeling. So homology modeling, okay, we are going to see Swiss modeler as an example. So virtually, so these are the steps happening when you try to feed a sequence for getting a structure. When we have to go for this computational prediction methods, if you see the computational prediction methods, this is the first method, homology modeling. So we have to go to this method if you don't have any 3D structures, uh, 3D structures deposited from gold standard techniques. So if you see the PDB database, we have around 2.32 lakh structures derived from gold standard techniques and 10 point so around 11 lakh structures predicted from computational techniques. So 11 plus 2 around 13 to 14 lakh structures. We have only 13 to 14 lakh structures in the largest database PDB. But if you see the sequences, I mentioned you we have more than 100 million protein sequences. 100 million. If you calculate in lakhs, then you multiply 100 into 10, isn't it? So that much protein sequences we have in the database. Just because of lack of technology, so we have very less structures. But if you want to work with the structures, uh, if then if the structures are not there in the database, one can go for computational prediction. So one such technique is homology modeling, which is very easier for us. Like if you have a sequence alone for the structure is which is not found in the database, we have to simply paste that sequence in the software called Swiss Model, an online server. Okay, after pasting this, we have a step or sub-step called search for templates. So our input is just to feed a sequence, like from a sequencer. Okay, if you are getting a sequence from a sequencer, just paste the sequence here in the window and you just search for templates. If you search for templates, you get an idea like what sample, what protein, and what species. Okay, so you also get an idea, and you can also select an appropriate template to build a model. So, but if you don't want to use two-step method for prediction, but if you want to use predict a structure with a single step by skipping the intermediate step like search for templates, we have to click build model. After pasting a sequence, simply click a build model. That's it. You get a 3D structure for this unknown sequence. Whatever sequence you have, you don't know what is the protein and you want to get a 3D structure, simply paste the sequence, click build model. So through the target sequence, and it just fetches a best template PDB. Okay, template PDB is the structure which is closely related to the target sequence. So the target sequence and template PDB is going to lead you a new model of a protein. Okay, so after predicting a model, so like when searching for a template, you get so many PDB structures like this. Whatever present in the top is more appropriate, is more close, resembling with the sequence. The structure is more, just because the structure is closely related to the sequence, one can click build models. Okay, likewise, I have ITSR. Okay, the ITSR is actually works on threading concept. The second method is threading. So according to the threading concept, one can able to understand that uh, in homology modeling through Swiss modeler, we are using one target sequence and one template PDB, isn't it? We use only two files to predict a structure for an unknown sequence. But whereas an ITSR, which uses threading concept, it uses multiple parent PDB files. For example, if you have a protein, participants, am I audible to you? Just interact with me. I want to check internet is live or
Yes,
not.
sir.
Yes, sir.
Good, thank you. Okay, if you have a protein of 110 amino acids, okay, of an insulin, like it's not a human, but it's some other species where the structure is not known. If you have 110 amino acids, I can have like for a first 30 amino acids. So if I segregate like 31 to 30 and 31 to 60 and 61 to 110, think like I have segregated into three different domains. So for each and every domain, I can have some 10 PDB structures. For this 10 and for this we have 10 more, we have for this 10 more. So totally 10 plus 10 plus 10, you have 30 structures. So but from the 10 structures, so whatever sequence you feed, it not only just searches the 10, that is the 10 PDB files which is having the 30 characters, that is which is having all the 30 is matching with the structures. You got it. First, whatever I give a sequence, okay, it splits into multiple domains, and each and every domain is going to search with PDB structures present in database. It takes the best 10. It takes the best 10 optimal alignment, the 3D structures. And from the best 10, it just screens the best one. Okay, finally, it happens in the multiple steps actually. First, it takes 10 and it takes only one. And here it takes 10 and it takes only one from the 10. And from 61 to 110 amino acid position, it takes the 10 and it takes the best one. So whatever you have, okay, you have small threads. If you have a small threads, so you are intertwining it to get a single thread. So the single thread is what I am telling as a final PDB structure. Just because we are using a threading concept like taking a multiple domains and predicting a best kind of a protein PDB for individual fragments. Okay, we call this as a threading concept. Okay, of course we use the lowest energy for for finalizing the best PDB file. So first we see how to work with the Swiss modeler. Target sequence is our design. Very good. And template PDB is predicted from the database with the target sequence, whatever you paste. I just show you with the Swiss model, you have some kind of a clarity. If I'm not showing you, I want to have a clarity. So just going with the Swiss model. So try to go with a sequence for which you don't have a structure. For this, I go with UniProt. So UniProt, I just go and take a protein. The protein what I use is a trap1. You know, in a trap1, human you have a 3D structure, but you don't have a structure for trap1 mouse. I simply click the trap1 mouse. Trap1 is actually heat shock protein sound effect. You click the structure, we don't have a structure. Okay. So just because we don't have a structure, I going to, but this is an AlphaFold structure. We don't have a wet lab I mean, the structure established through wet lab technique. So I'm going to use it. You can use even the AlphaFold, but if you want to use a Swiss modeler predictor protein, one can take a sequence. I just take a sequence now. The sequence is around 706 in natural amino acid protein. But still, just because we want to predict a structure, I just copy it. You can either copy the amino acid sequence alone, or even if you copy and paste the FASTA format, okay. So I just going to the Swiss model. It goes to the swissmodel.express.org. One has to click start modeling. This is the first step. And paste the FASTA format. Even though we paste the FASTA format, it takes only the amino acid sequence. So whatever we pasted, okay, just because the prediction takes up to 5 minutes or more than that, okay, I suggest you to give a name for the project. If you are not seeing the result on today, on tomorrow, the mail, this will be generated in, this will be sent to my mail. I can show it from the mail. So I can also provide my email ID. So this is the project title, trap1 protein of mice, isn't it? So I gave like trap1 underscore mice. Simply, this is the intermediate step I'm going to try at present. Search for templates. Okay, so it's going to search so many steps are happening in background. Okay, whatever steps are happening in background, I showed you as the steps earlier. 6 steps are there. So what is going to happen to running HHP leads against SMTL, a kind of a language, catch query alignment, predicting this. So this is all a virtual thing happening. But what is the final template you get and how to predict the model? It takes some time. So meanwhile, I can show you something on the other server. What I mentioned is ITSL, isn't it? Which uses the
Sir,
threading. Tell me.
as sir, in this case, if we directly click on the build model, it would
No, no.
consistently
Excuse me,
build a
excuse
protein
me.
model.
No, your voice is intermittent. Actually, your voice is not continuous. Can you repeat once again?
No, sir. Instead of search for template, if we click on the build model, it will
It definitely gives you the result in a single step.
protein
If you
model.
click, yes, if you click build model, you get in a single step. But if you want to be confident on what template you are searching, you are choosing, okay, one can do this because in getting a template, we get a kind of a result like, so see this template, how you get, here you find like identity, 100 percentage. Like, for example, we have, first we have to see the protein. If you have a protein with 830 amino acid, and the first template is having just 400 amino acid, and 400 amino acid of template is matching with 400 out of 830 amino acid of our query sequence. Then, but if you see the second protein, which is having 500 amino acid coverage, okay, but you can see 450 amino acids, 450 amino acids, or matching with the 830 amino acid protein. Then instead of 400, all the 400 matches, but here you have 450 matches, 50 mismatches, but still you have 450 matches, which is greater than this, the previous CT, isn't it? So one can choose the 450 matches protein sequence, the second template. Instead of first template, you can also choose the second template. So based on your choice. But most probably the first hit gives you the best template. We can check that also. Okay. If you see the identity and coverage, one can decide, okay, even what template can be chosen. Okay. So this is a determining step. Okay. Like coverage and identity. Only through coverage and identity, only we have to finally decide. Any other doubts, sir? Subhajit, sir? You have any doubts?
No,
Is
sir.
it clarified? Okay. So only
Yes,
thing is,
yes.
this is the intermediate step. It takes some time to get. So meanwhile, I can go to the ITSF. So the name itself, here I can show you the iterative threading assembly refinement. So these are the sub steps happening inside. Iterative threading assembly refinement. That's what ITSF. Okay. The expansion of ITSF. If you see the ITSR, okay, already 7.94 lakh structures are predicted from this. So one has to copy the protein FASTA format of the protein sequence. But here it is mandatory to initially register. One has to register and then you have to log in. Okay. So email is mandatory here, but whereas in Swiss model, there is no mandatory. Here you have to provide email and you have to provide the password also. So one has to initially register and you have to log in to work with the ITF. That alone you have to take care. Okay. And we have plenty of tools. Oh, okay, now I got a template. See, in this templates, okay, so the first one is showing something like identity 78.53. The second is showing 78.02. But how about the coverage? The coverage, are we getting something? Here we have coverage of 70 to 704. If you keep your cursor, you are getting some information. This is chain B 72 to 704. Coverage is 0.96. Here the coverage is also 0.96. Same coverage, but the percentage of identity is more in the first hit. So the first hit is the default hit. Sometimes even the coverage more is also present in second one. So one instead of choosing one, first one, you deselect this and click the second. In our case, default we had the first hit as the best. So simply going to click build models. So this is the second step. So you can also build models without checking, but this gives a clarity. Okay, we'll be getting a structure soon. So whatever structure we get, we have to download the structure in the form of PDB format. And that structure can be visualized through a visualizer called Discovery Studio. So I've already downloaded structures. Okay, so if you want to download a structure, okay, and after downloading the structure, if you want to visualize it, open with BioVia Discovery Studio. Give me 1 or 2 minutes to complete. So I'm just going to show you how the protein structure can be visualized. So we have plenty of visualizers to visualize the protein. Discovery Studio Visualizer, RASMOL, PDB Viewer, Python Molecular Viewer, TN3D, Java Molecular Viewer. Likewise, we have plenty of molecular viewers, both in offline and online. Okay, Chimera, etc. So whatever visualizers we use, okay, but I mean, there is a no need to use all the visualizers. There is a no, what is a necessity to go with a command line kind of a visualizer if you can able to interact everything with mouse click, mouse left click, right click, drag, mouse drag. So everything you can interact with the mouse events. This is very easier for us, okay, to interact with Discovery Studio Visualizer. So ignore the other visualizers, whatever I mentioned. If you want to analyze a talking result, or if you want to model, or even if you want to find the binding sites, this is the best visualizer and this is the freeware. Anyone can go to the BioVia website and download this Discovery Studio Visualizer. So in this protein, the insulin protein, whatever you find in the red color are nothing but alpha helices. And the beta sheets are shown in blue color strands. And the loops are generally shown like this in gray color. In most of the visualizers, we find like this, including Discovery Studio Visualizer. We find so many kind of options here. So one can go to the BioVia website and download this visualizer. A protein looks like this. Now let me open the result. So we got a nice result of a PDB. So today I'll be downloading this file. So here if you see, we got only one model. So I click download files, and if you click PDB format, okay. So I'm going to download this model 01 in the desktop on the bioinformatics demo. So whatever I downloaded, okay, just open this and more about the structure, validation, whatever we predicted, we discuss it on tomorrow. So whatever model I predicted for the trap1 looks like this. See, for human insulin, we have almost a single chain. But for the model trap1, we have multiple chains. Multiple domains are shown, isn't it? So we have whatever blue color is the beta sheet. So this is one domain, second domain, third domain, fourth domain, five domain. We have plenty of domains in the beta itself. And how about the alpha? Still we find multiple. Okay, it's the largest protein because we have 830 amino acids, isn't it? So that's such a kind of protein we have generated. Okay, so participants. So today I've completed my session on protein structure prediction. Tomorrow we discuss the left out part. Okay. 3D structure, the model, whatever I download has a 3D structure. Whatever I showed is the 3D structure of the model. Okay. Any other doubts you have? Fiery is different. Fiery is something like used for visual screening. That's not a visualizer. If you want to visualize, you have to screen the compounds through binding. One can use the no. Fiery, yeah. Fiery software is threading server for protein structure prediction. So you can either use Swiss model or Fiery. Okay, so if you want threading as the best, the threading gives you the best structure, but it takes more time to generate the result. So in Swiss model, we got within 5 to 10 minutes, but in threading, you may get even more than 1 day to get a PTV. But if you want to complete the result first, one can use the Swiss modeler and convert to Fiery. Okay. Okay, participants. Thank you for your interactions. So tomorrow we discuss the